When you walk in a supermarket, a security officer will check your temperature using a thermometer gun to see whether you're infected by COVID-19 or not. However, this method is not accurate at all. COVID has an incubation period of 5 to 6 days, but it can go up to 14 days. This means you can never know if you're infected in the first few days. Even when the symptoms starts to show, how would you know if you get COVID? Or it is just like any other common cold? Or when you just exercised and your body temperature goes up? Well, if you have a high temperature because of that, well, that doesn't mean you get infected. So, how do you get tests for coronavirus and how do they work? The first way for testing that I want to talk about is a PCR test. You sample with nasal swab to collect mucus deep in your nose because that's where the virus lives. Here is a diagram of the coronavirus. As you can see here, it has an RNA surrounded with fatty membranes and it is covered with spike proteins. These RNA are like DNA, but they are only single-stranded. So, from the sample, we extract the RNA and copy that into DNA through a process called reverse transcription. Once we have the DNA, we put it into a PCR reaction. Here, DNA is denatured to split it into two separate strands. Then, these primers that matches to the DNA binds with it. The DNA polymerase sorts of zips up the DNA strand, which leaves us with two exact DNA copies. And then you just simply repeat this process multiple times until you have billions of copies. Finally, you can see whether your sample looks like COVID-19 or not. And next, we have the antigen test. Similar to the PCR test, you collect samples through nasal swabs. But unlike the PCR test, instead of testing for viral RNA, this time we detect pieces or antigens of the virus, like proteins that surrounds the coronavirus on the diagram I showed you before. Once we got the sample, it is reacted to expose the antigen. After a while, the sample is placed on a testing strip, as you can see on the picture. The particles will move along the testing strip by using capillary effect. In the conjugate pad, the particles bind with antibodies labeled with fluorescence that will make it light up. Then the particles move to the test line. The test line here is covered with immobile anti-COVID antibodies. If the antigen is present, the test line will light up. But if there are no antigen, well, the test line will not light up. And then finally, the control line. It is what decides if the test is valid or not. It binds with the non-target antigens, and if it does, it will light up. So, for the results of this test, if both the control line and the test line light up, then you are tested positive. But if only the control line lights up, then you are negative. However, if neither the control nor the T line lights up, or only the test line lights up, then it is invalid. So you should do the test again. The last test is the antibody test. It detects presence of antibodies in your blood, or more specifically, IgM and IgG. So when the body is infected, it responds by producing IgM antibodies for a quick response. The second wave of response, the body produces IgG antibodies that provide a longer lasting immunity. As you can see on the graph right here, after the incubation period is over, your body starts to make a response, yeah? So for the first 7 days, your body produces IgM to fight off the virus. And that's why it's called the rapid response. But then after 7 days, your body starts produces IgG antibodies. You can see on the graph there that unlike the IgM that diminishes after a month, the IgG stays for a long period of time, even after years, so that when you get reinfected again, it is still there and ready to fight back. Now, there are two types of antibody tests, the rapid antibody test and the ECLIA serology test. The second one is only recently developed, so it's okay if you haven't heard of it. So, the rapid antibody test works the same way as the antigen test. If you look at the picture, you might notice that it's almost exactly the same with the antigen test. So, blood sample is placed on the sample pad where it will flow through the testing kit. On the conjugate pad, IgM and IgG binds to gold COVID-19 antigen conjugate. On the M line, IgM binds to anti-human IgM antibody. While on the G line, IgG binds to anti-human IgG antibody. So all of these places, they have specific anti-antibodies for them to bind to. And finally, on the control line, there are gold rabbit IgG conjugate that binds with anti-rabbit IgG antibody to validate the results. In electrochemiluminescence immunoassay, or ECLIA for short, proteins that match COVID are produced, and then they are labeled with ruthenium that acts as a biological luminescence, and biotin that acts as an anchor. These are then incubated with the blood sample. In the blood, if antibody is present, they will bind to the antigen. However, if there are none, then it will not. Then, tiny magnet beads bind to the biotin. They are then placed on an analyzer, as you can see in the picture, where magnetic current is applied so that the magnet sticks to it. After that, a solution oxidizes the ruthenium and it produces light. The brighter the light, the more antibodies are present in the blood. And so this is a conclusion of all four COVID-19 tests: the PCR test, antigen test, rapid antibody test, and the ECLIA serology test. If you look at the table, you might notice there are differences between the tests, and this might help you decide which test you should take. First things first, the PCR test. It is 15 to 30 minutes long, and the results are out in 1 to 7 days, and it is the longest time needed for a test to conduct compared to the rest. The PCR test collects sample from the nasal mucus, and it detects the virus genetic material, which is the RNA. The best time to test PCR is 5 to 7 days after exposure to the virus. If it is positive, then it means you have an active virus infection and you should isolate immediately. The pros of this is that it's highly sensitive and specific. It can detect up to only one, just a single virus, and you can immediately know. However, the cons are it's expensive and you have to do it in the lab, and not all labs are capable of doing a PCR test. Next is the antigen test. It is 15 to 30 minutes, just the same like the PCR, and the results are out in 1 hour to a day. So it's faster rather than the PCR test. The sample collected, just like the PCR test, are through nasal mucus, but unlike it detects viral antigen instead of RNA. The best time to test is 1 to 5 days from onset of symptoms. If it is positive, that means you have an active virus infection, and just like PCR, you should isolate immediately. The pros of this is that positive results are usually accurate. However, the cons are negative results should be further tested with PCR. On the rapid antibody test, the duration is only 10 to 15 minutes, while the results are out in 30 minutes to a day. The rapid antibody test is the fastest test that we have right now. In this test, samples are collected from the blood of your fingertips and it detects IgM and IgG antibodies. The best time to test this is 7 days after exposure to virus, where if you remember the graph before, it is when IgM and IgG are produced by the body. The pros of rapid antibody test is that it's cheap, widespread, and easy to use. Even you can do it at home. I'm pretty sure you can buy a rapid antibody test kit online and then you can test it yourself at home. However, it is very low accuracy with many factors, so that like time and then maybe the kit like that. It doesn't really tell you whether or not you're infected, but more like it says whether you've had an active infection in the past or not. And the last one is a clear serology test. This one is also quite quick. The test duration is only 10 to 15 minutes, and however, the results are out in 1 to 2 days. And this is due to the fact that you have to do this in the lab and you have to wait for the incubation period of the blood. This blood, just like the antibody test, it detects IgM and IgG antibodies. The best time to test this is 7 days after exposure to virus to make sure that you do have antibodies. The positive results mean that you've been infected in the past. The pros is that it's way more accurate than rapid antibody test. So this time you are actually sure whether you've ever had an infection or not. However, a clear serology test, it's still a relatively new technique. That's why it's not so much widespread, and also the fact that you have to do it in a lab. And there we go. Thank you for listening. I hope everyone stays safe in these trying times.